All right, so now that we've talked about pH, let's talk about some applications of pH, and that's related to buffers and titration. So make sure you know kind of how these things relate to pH, what their definitions are, what they're made out of, that sort of thing. So a buffer is a substance that helps keep pH the same or make a consistent level. All right, so if you want your pH to be 8, you're going to add a particular buffer that will help counteract any changes, any extra additions of an acid or a base. Or if you want your pH to stay 5, a different sort of buffer would work at that level. All right, so buffers work by keeping your pH level the same or very minimal changes. Buffers are made out of a weak acid and a weak base, or essentially a weak acid and its conjugate base. So what that means is you've got your acid, all right, which, skipping down to this question down here, what adds the H+? What gives off the H+? Is it the acid or the base? Think back to your definitions. Arrhenius or Bronsted-Lowry will come in handy here. Hopefully you were saying acid. All right, so your weak acid is going to have an H+ it can give away, and when it gives away that H+, the molecule without the hydrogen on it, that's the conjugate base. So I'll have an example of this on the next slide so you can see that. But essentially you have an acid and a base in the buffer, so that whether you add an acid, you have a base to neutralize it. If you add a base, you have an acid to neutralize it. So a buffer has both pieces so that it can neutralize anything that you add to it. So a good example of this is your blood. All right, humans need their blood to be at a pH of 7.4. So is that acidic or is that basic? So we're just barely above 7. 7's neutral. All right, and slightly above it means that our blood is slightly basic. All right, so and our blood carries this compound called carbonic acid. All right, so the carbon dioxide that we breathe out in the blood actually doesn't usually travel as pure CO2. It gets converted into this compound called carbonic acid. And carbonic acid is an acid. All right, and it can give up its proton to become a proton and bicarbonate. All right. So out of this, so we know that carbonic acid, I mean, kind of no-brainer, that's our acid. Which of these two, the hydrogen ion or the bicarbonate, is the conjugate base? All right. So remember, base is going to be the one that can accept that proton, can pick it up. So hopefully you are thinking this one. So all right. So we've got our acid, which is the carbonic acid, and then our conjugate base, which is the bicarbonate. All right, so this is just an example of where a buffer comes in handy and works for things. So you've got this acid that has an extra proton it can give away if we add a base to the blood. You've got this base that can pick up a proton if we add an acid to the base, to our blood. Sorry. All right, so buffers work to keep the pH the same by having a weak acid and a weak base available to neutralize whatever we might add to it. You know, if you add too much, these won't work. But small extra amounts of acid or base, like if you have some lemonade which has lemon juice, all right, or you have some bitter cranberries or something like that. So our bodies can deal with us eating acids and bases and things like that. All right, so the other thing we need to focus in on is titration, and this very much ties into your lab. We've already discussed this a little bit, but titration is a way of kind of working backwards to figure out what the pH of your solution was. All right, so if you don't know what the solution is, if you are given an unknown and you have to determine its starting pH, or you are in a lab and you accidentally add more acid to your solution than you think you were supposed to, you can double check it with the titration. Okay? How this works, so titration requires the use of an indicator. All right, so in your lab, we had the phenolphthalein indicator. We also had the alkalinity indicator. All right, so there's the phenolphthalein goes from pink to clear at 8.3, and then the other indicator went from, I think it was green to red at 4.5, something like that. All right, but these indicators have a specific pH where their color changes. And if you know how many drops it takes to cause that color change, you can work it backwards to figure out what your starting pH and starting concentration of hydrogen ions was. All right. So for this example here, we're going to focus in on phenolphthalein. All right. So these two vials, A and B, all I've done is added phenolphthalein. All right. So based on that, what can you tell me about these two vials? All right, so, and I gave you a little hint here. Remember that the phenolphthalein changes color. It goes, when you hit 8.3, it goes from pink to clear. So what can you tell me about these two vials? So I'm hoping what you are saying is that vial A, pH has to be greater than 8.3. All right, because phenolphthalein goes clear once the pH drops below 8.3. All right, and B, you've got to tell me that without any more information, really all you can say is that maybe pH is 8.3 or lower. All right, if I told you that I had added, I had just added 5 drops and after the 5th drop it went clear, then you probably could say it was at 8.3. But you don't know that. All you know is that you added phenolphthalein and it was clear. So it's got to be 8.3 or lower. All right, so that's one way you can use your indicators. Titrations can, and luckily we are not going to get into this detail with this in class here, but if you remember stoichiometry from Unit 6 in Chemistry A, you can, once you know how many drops of your known acid or base that you add that causes the change in your indicator, you can work that backwards to figure out what your concentration of your unknown substance was. All right, so, but luckily for you, we're not getting into that crazy math. Instead, we're going to use more of a relative sense. Then we can tell, approximately what our pH should have been, especially if we're comparing two samples and they require a different number of drops. You can relatively figure out, which one was more acidic or more basic at the start. So, for example, you're doing this in your lab. So you add drops of acid to 2 solutions until the phenolphthalein indicator goes clear. So, first question to you, are your solutions starting out as acidic or basic, and how do you know? So think this one through for a second. This right here should be enough on its own for you to kind of think about this. So we're going clear, which means we're starting out not clear. So which means we're starting out above 8.3. So our solutions were basic at the start. All right? And when it changes, they're still basic, it's just they were more basic. So that's the first key to kind of understanding what's going on in your lab. The next thing is, you can also think of it in terms of, if we're adding an acid, then we're dropping the pH, so we had to start out at a higher one. The next thing you can do is comparing the number of drops. If it takes 5 drops for solution 1 to go from pink to clear, and 12 drops for solution 2, which solution had the higher pH at the beginning? All right, so again, focus on the fact we are adding acid to this. One of these requires more acid to get the pH to fall to 8.3. So I'm hoping you are thinking that solution 2 was the more basic at the start. And think about how you could explain why. Alright, the why is because more acid was needed to get to the same pH. Alright, it took 7 more drops of acid to get us to the color change. When we reach that color change, both solutions are at the same pH. The only difference is that the solution 2 required more drops of acid to get there. So that means solution 2 had to have more base to begin with. Hopefully that makes sense. But if it doesn't, come get help. We'll talk about this again when we go over the lab report too. But if this is already making sense, that is wonderful.